So let's talk about raw material. What's raw materials like for a vaccine?
So it's funny calling it raw material, but you're right, it is. Antigen antigen is the main material. So in immunology, an antigen is a molecule that binds to a specific antibody or T cell receptor. So essentially binds to your cells inside.
Hmm.
And the presence of this antigen may cause your body to trigger an immune response. So, immune response, you're fighting something off, right?
Hmm.
And antigens can be proteins, it can be polysaccharides, it can be lipids, it can be nucleic acids, it can be whatever. And this antigen could be inside of viruses, could be inside of bacteria, it could be inside of whatever host that the scientists determined that it will be a great place to grow. Okay, so first, it's a painstakingly long process for the scientists to understand what the antigen is, how do they grow it, what could affect what, and there's a lot of research that goes into this.
So the
Then
antigen is the way that it sounds like that's how you deliver the virus into the body. It's like a vehicle.
the virus is the vehicle. For example, the virus or the bacteria is the
Mhm.
vehicle, is
Yeah,
the host, and what's inside of it, the DNA, the
yeah.
receptor mechanism, whatever, that is the antigen that binds to the receptor in your cell.
Okay,
And then it creates replicating of the DNA of the
got
good
it.
stuff that
Got
you
it.
want, and then that helps you fight it. If I step back, so I want to just explain how these viruses or even monkeypox kind of affects you, and I briefly touched on it. So once it targets your immune cells,
Hmm.
right, binds to them, uses its cell mechanism to replicate its own DNA, engulfs it, exits out. As it exits, it bursts. Essentially, it's called apoptosis, which is cell death.
Hmm.
So, and then it does keeps doing this. So this is why your immune system goes into this like super shock, flashes, rash, fever, all flu-like, and so it's causing cell death, which is why it's these viruses are their mechanism to do this is amazing, because on its own it can't do anything. It's using your cells to do the damage.
Yeah.
Okay, so let's go back. You got your antigen, you know what you're going to use. Cool. Now you got to isolate it from whatever you grew it in, right? So you grew it in a, let's say you grew it in a bacteria. You got to take your antigen out, purify it. So what they I'll talk about what that means in a bit, but walk with me here. Free from
Hmm.
contaminants, you got your antigen now. All alone, solo mission, antigen is ready. You go
Hmm.
into formulation. So that pure antigen is mixed in with other ingredients for your final vaccine. I'll talk about what those other ingredients could be in a bit. But okay, now you have your final vaccine. You go through a series of testing. So first is preclinical trials. What they'll do is they'll take the vaccine, they will do all sorts of lab testing, property testing. They will trigger immune response from a chemical perspective. They'll see, okay, what is this? What is it going to do? How does it work? And then it moves on to animal testing. So depending on what the best animal for testing, it could be a rat, it could be a hamster, it could be a primate, whatever, right? They will do animal testing. Once they get the green light from animal testing, they go into clinical trials, so human trials. There's a lot of phases. I'm not going to go through the phases, but essentially, human testing has small scale to large scale. They get the approval from there. It goes into regulatory review and testing. So regulatory review, so agencies like the FDA for the US, Health Canada, well, for Canada, EMA for Europe. They look at this, they go through all the documentation, they go through the vaccine, they do their own testing, and then you get approval. And then the manufacturing actually starts.
This is the approval. Yeah. So it goes through this rigorous process to essentially say, look, my vaccine not only does it work, it is repeatable and it's safe.
Correct,
And this
correct.
is part of the disputes, if you will, or concerns that a lot of people had about COVID vaccines when they first came out, because they were accelerated and they had to skip through
Yeah.
some of those steps to get it to market very quickly. And that's what a lot of people were like concerned about, like how, because that process alone takes years for any vaccine to
Yeah.
be completely
Yeah, it could
developed
take
under.
years.
Yeah.
And I remember that here too, right? In Canada, that was a major point of discussion, is how could they do something like vaccine from nothing to something that fast?
It was within 6 months, I believe. I remember, if I remember
Yeah,
correctly.
it
It
is.
was
It
very
was
fast.
rapid. Now,
Yeah.
again, these pharma companies have so much resources at their disposal, and if the government is also throwing resources at it, who knows what they can do? But that point is still valid. Is they did it so fast. It is
Yeah.
not normal, and I get it. And we could talk about that all we want, but it is what it is.
Okay, so now that we have the approval of the vaccine and we got FDA approval and we're good to go, how do we manufacture them on this massive scale that we're able to for the entire population?
Bioreactors are used to grow the culture medium. So when I say culture medium, this is the antigen and this is the host that I talked about. So host, bacteria, could be virus, and the antigen is inside of it, right? Bioreactor, think of a giant tank.
Mm-hmm.
They put the culture medium inside of it. They give the tank the right amount of oxygen. They give the tank the right amount of nutrients. They give humidity. They control humidity. They make it a best
It's like a
environment for
petri
this
dish.
bacteria.
It sounds like a petri
Yeah,
dish on an industrial scale.
that's a great analogy. Yes, petri dish, large scale, big tank, stuff is growing, and this could take several days. This could take a few weeks. What it depends on what the culture is, what bacteria they're growing, what facility capabilities are. Few days to a few weeks. So now you have your culture. All right. They use giant centrifuges. So if you remember your science class, a centrifuge, you put your little sample vial in one side, you put blank vial in the other side, and it spins really, 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 really fast and separates the components
Yep.
based on the heaviness or densities.
Mm-hmm.
They do the same thing, but on industrial scale. They use large centrifuges to take that culture medium and they spin the crap out of it to get the components to separate. So antigens and its host is separating from all the other culture medium, all the other stuff. It could take up to 60 minutes for this action to complete. Then they use chromatography, so industrial scale chromatography, to remove all the impurities and to isolate the antigen. So now you only get the antigen. It takes a few hours to a few days to go through the chromatography method to get your antigen at the end. That product is the washed or purified antigen. That is the raw material for everything to come now.
Okay, that's
Yeah.
so that you do that cooking process, if you will, so that you can create the antigen that you need for the specific virus, and then you separate
Yes.
it out of the other ingredients into its own.
Correct.
Now you have
Correct.
your custom-made antigen. What do you do with
Correct.
that, and what are the next steps?
All right. So antigen, and I also mentioned other ingredients, right? So large mixing tanks, they add the antigen, they add stabilizers. So stabilizers are amino acids, which is the building blocks of life, I guess.
Mm-hmm.
Proteins, sugars, so glucose, could be fructose, could be whatever, right? Preservatives. Thiomersal is often used in vaccines as a specific preservative because you want to prohibit bacterial fungal growth in your vaccine. And then diluent. So saline, the IV drip saline?
Yeah.
Saline.
They add
Think
that.
of
Okay.
it, yeah, think of it like a buffer solution in your vaccine. Saline. And so all this is mixed. And remember that this is all happening in a sterile environment, aseptic environment. And the mixing actually takes 15 minutes to up to 2 hours. And so it does have, agitation because you're mixing all this stuff in. You got to have a homogeneous mixture. And that final mix is your vaccine.